Hello everyone and welcome back to Microbial Concepts. So in this video, we are going to discuss about identification of unknown bacterial cultures. So yes, you are already introduced to Bergey's manual of determinative bacteriology, say from your first year. So that's what we use for identification. So I'm going to discuss steps like how to proceed for identification of a given bacterial culture. Okay, because I got this request in a comment box saying please make a video on identification of unknown bacterial culture. So this is the video. Do watch it till the end to clear your concepts. Okay, so let's start with the video. Yeah, so our objective here is to identify a bacterial unknown culture in, say, from a mixed culture, or say you got it in isolation from a given particular sample and you want to identify it. Okay, so that is our objective, and we use Bergey's manual. We use biochemical test to identify. Till college level we identify till the genus. Okay, and hardly species identification is done in your colleges. So it is okay if you have identification till genus, but in your masters you may require to identify till the species level for your projects, for your dissertation. Okay, so I'm also going to discuss that. So to watch this video till the end. Okay, so the identification of bacteria is done using many different techniques to narrow down the type of bacteria that are present in your unknown bacterial culture. Okay, so now your culture can be, say, pure single. Okay, means after streaking or after doing a spread plate you get a single type of colony. Okay, that can be a case, or you get a mixed culture. That means you are having 3 to 4 different types of cultures growing. For example, let's assume a practical or say experiment of isolation of starch degrading bacteria. So when you use soil sample for such kind of experiments, you get 10 to 20 different cultures which are able to degrade starch, okay, or hydrolyze starch. Now in that case you need to identify all those, and then you start studying them. Okay, like, can I optimize this particular culture? Can I improve the strain and get more yield and all? Okay, so that's how our experiments are, and that's the reason why we need to identify our culture. Okay, and also after identification we need to check that whether this particular culture that I am handling is a pathogen or not. Okay, because till it is unknown you don't know anything about the culture that it is a pathogen or not. So while handling any given unknown culture, please follow some precautions so that you don't get any infection. Okay, so now first thing when it comes to any experiment in microbiology is to isolate a pure culture. Okay, so the first important step is to isolate pure colonies of bacteria. Now the streak plate method is widely used method, but even pour plate or spread plate is also used. That depends on your type of experiment. Now already a video is uploaded on streak plate method. Okay, you can watch that. And also a video is uploaded on colony morphology and colony characteristics. Okay, so how to write report of colony characteristics or colony morphology, everything is discussed in this video. So do watch these both these videos if you are not clear with the streak plate method or colony characteristics. Okay, so once the isolation is done, what we do, we perform colony characteristics, we note down our results, then we perform gram staining and motility. Okay, and then we move further for Bergey's manual for identification. Now we will take an example here, like for any XYZ experiment we want to isolate and we want to identify a culture. So starting from isolation, what we will do, we will add 1 gram of soil in, or 1 ml of water in 9 ml of saline suspension. We will mix it and we will keep it undisturbed for some time. Then the supernatant is what we use for isolation. Now you can perform serial dilution and you can do spread plate technique or say pour plate technique, or you can directly go for loop full of the sample with streak plate. Okay, now while you are performing these techniques, isolation techniques, you need to follow aseptic techniques to avoid contamination. Okay, It is easy to identify contamination in streak plate, because if your colony is growing out of your streak line, then it is a contamination. But in case of spread plate technique, where you are spreading using a glass spreader and colonies, they are growing randomly here and there, it is difficult to identify the contamination. Okay, so do follow aseptic techniques and try to avoid contamination. Okay, so once you have isolation, you mark a fully grown mature colony, bacterial colony. You note down colony characteristics, you perform gram staining motility, and then we refer to Bergey's manual of determinative bacteriology for biochemicals. So this is how it looks. So mostly ninth edition is, I guess, present in every institute, every college, and it is what is referred. So there are two types of Bergey's manual. One is determinative bacteriology and another one is systematic bacteriology. Okay, so systematic bacteriology is actually for classification, and more descriptive information is given on each genera and each species of a bacterial culture. Okay, but determinative bacteriology is not for classification or any taxonomical study. It is just for identification. Okay, so Bergey's Manual of Determinative Bacteriology is for those who are primarily engaged in identification and they are not concerned with the classification. For those who want to identify a culture, they should use determinative bacteriology. Okay, so it has no descriptive info, that is information on genera or species, like systematic bacteriology. Okay, so there is difference between determinative bacteriology and systematic bacteriology. There are two types of Bergey's manual. Don't get confused. You can have a question in your Viva like which Bergey's manual is used for identification. So it is determinative bacteriology. Okay. Now let's move further. So about determinative bacteriology, I am going to discuss few points here which I think are important. If you are new to Bergey's manual, if you have not handled Bergey's manual before, then you should know what this book is. Okay, so first chapter, there are different chapters, 4 5 chapters I guess in this book, and then there is all the description given about the different groups, different categories of bacteria. Now the first chapter is about using this manual. Okay, so this description is what I have given here in short. Now about the general approach of using this manual, how to use this manual. So the first is the nature of bacterial identification schemes. Okay, so it is important to know that this book is mostly used for identification purpose. It has nothing to do with the classification scheme. Okay, so the features that are used to differentiate various organisms, they often have little to do with the fundamental basis for arranging the organism into taxonomic group. Yes, the very little of the fundamentals of identification are also related to the taxonomic group, but very little. Okay, so this book is purely for the identification purpose. Second step. Now, before using Bergey's manual, you should know that my isolate is a prokaryotic microorganism or a eukaryotic microorganism. Why? Because this is Bergey's Manual of Determinative Bacteriology, that means study of bacteria. Now bacteria, they are prokaryotes, they are not eukaryotes, so you will get only prokaryote information in this Bergey's manual. So if you are dealing with say fungi or algae or yeast, okay, then that comes under eukaryotes and Bergey's manual is not used for them. Okay, then third step is to which major category of bacteria does my isolate belong. Now there are main four categories of bacteria, and they are described as gram negative U bacteria that have wall, gram positive U bacteria that have cell wall, then U bacteria which lack cell wall, and archaeobacteria. Okay, so now even prokaryotes, they are grouped as U bacteria and archaeobacteria. Now archaeobacteria, one which um, which live in very harsh or extreme environments. Okay, and eubacteria are the bacteria which are widely found here and there. Okay, simply just to cover this. Now, even a video is uploaded on this particular topic, that is difference between prokaryote, eukaryote, then difference between the um, archaebacteria, okay, and eubacteria, the economic importance of archaebacteria, everything is discussed. So you can find these videos on my channel. Okay, so now let's move further. So after this, once you have identified that to which category my bacteria belongs to, then next is to which group does my isolate belongs. Okay, so to which group, after categories there are groups. Okay, so these four categories, they have around 35 groups. Okay, so you need to do gram staining motility, and after that you can identify to which particular group my isolate belongs to. Okay, so once your group has been identified, you start performing biochemical test so that you can find which genus does my isolate belongs to. And once your genus is identified, you can find out to which species does my isolate belongs to. Okay, so this is how you have to step by step use this particular manual. Okay, then next is the four major categories of bacteria, so that I have already discussed. That is, the first is gram negative U bacteria, which has cell wall, gram positive U bacteria that has cell wall, then U bacteria which lack cell walls, and fourth is archaeobacteria. Okay, so these four categories, they have around 35 groups. Okay, so we are going to study those. So you can see there is more detailed information about each particular group is also given. So there is a table you can see here. I am not going to discuss all those 35 groups important features because it will take too much time. So you can see the first group is, you can see, you can compare here, the contents here are given here. So you can see there are 5 chapters. After 5 chapters you get this index where first group is of spirochetes. So you can see their important features are in short mentioned here. Then next is aerobic, microaerophilic, motile, helical, vibroid, gram negative bacteria. This is the group second. Okay, so even the important features of this particular group is given. Okay, not just this, all 35 groups important features in short they are mentioned here. Okay, so if you have what I can say, confusion about deciding to which particular group my isolate belongs to, you can read these information, important features, and then you can decide. Okay, so you can see this is the content. First group is pyrochete, second is aerobic, microaerophilic, motile, helical, vibroid, gram negative bacteria, third is non-motile, gram negative curved bacteria, fourth is gram negative aerobic or microaerophilic rod shape cocci. Then comes facultatively anaerobic gram negative rods. So you can see below these groups the genus or the list of genus is also mentioned. Okay, so there are even some groups, they have subgroups. Okay, then sixth group is gram negative anaerobic straight curved and helical bacteria. Seventh is dissimilatory sulfate or sulfur reducing bacteria. Eighth group is anaerobic gram negative cocci. Ninth is rickettsia and chlamydia. 10th is anoxygenic phototrophic bacteria. 11th is oxygenic phototrophic bacteria. Then 12th is aerobic chemolithotrophic bacteria and associated organisms. Then budding or appendage bacteria. Then sheathed bacteria. Then 15th is non-photosynthetic, non-fruiting, gliding bacteria. Then 16th is myxobacteria, the fruiting and gliding bacteria. Then 17th is gram positive cocci. So you can see from gram negative to gram positive we have covered. Then further is gram positive rods and cocci which are endospore forming. Then group 19 is regular non-sporing gram positive rods. 20th group is irregular non-sporing gram positive rods. Then 21st is the Mycobacteria, which has only one genus. Then group 22 to 29, okay, they are all actinomycetids. Okay, different groups. Then group 30 is mycoplasmas which lack cell wall. Okay, so we are towards the third category. Okay, then the fourth is of archaeobacteria, that is methanogens, archaeal sulfate reducers, extreme halophiles, halophilic, aerobic archaeobacteria, then cell wall less archaeobacteria, and extremely thermophilic, hyperthermophilic metabolizers. Okay, so we have covered all four categories under 35 groups. So you can imagine the number of genus and number of species that is covered by this particular single book. Okay, so you just need to identify properly that to which category and which group my genus belongs to, and then you can perform biochemical test and you can identify your genus. Okay, so in first year you must have gone through such kind of charts. Okay, so these are charts which are made by the lab assistant or say by the colleges to simplify your work. Now in first year when you're introduced to Bergey's manual, by big seeing that particular big book, you may get confused. So to simplify the identification of a given bacterial culture, these simplified charts are made. Okay, they are similar to the approach that we are following by using the Bergey's manual, but here something is missing. What is that? So not each and every chart has detailed list of biochemicals present. Okay, so you can see here in case of gram positive bacteria, so you have a gram positive bacteria. It can be a bacilli, that means rod, it can be a cocci, or it can be a branching filament. In case of rod, it can be aerobic or non-aerobic. In case of aerobic, you can see just three examples are listed here. Okay, this is to just minimize your confusion so that you can easily identify your isolate in your first year. Okay, but after that you have to consider the whole lot of number of genera which are present under a group, and you have to specifically precisely identify your culture. You can't just rely on three or four examples, right? So in case of cocci aerobic, then that can be a streptococcus or staphylococcus. These are the generally used cultures in your college level, so those are listed here. So staphylococci or streptococci, then further you can do these biochemicals to identify and confirm that whether your culture is streptococcus or it is a staphylococcus. In case of branching filaments, aerobic, non-aerobic, so aerobic is mostly nocardia, that is actinomycete, and another non-acid fast actinomycetes, they are anaerobic. Okay, so depending on your gram positive result or gram staining result, which is gram positive, depending on the shape of your bacteria, you can easily follow this chart and you can identify. But I am again marking here my words that this is just for first year students, okay, who are new to Bergey's manual, and it is difficult for them to handle the book and follow these steps to identify. Then in case of gram negative bacteria, diplococci means two cocci. Cocobacilli looks like rod, but it is a cocci. Then rod shaped bacteria, which is also known as bacilli, and comma shaped. Okay, so you can get any of these different shape in your culture. So suppose you have a cocobacilli. So what are the cultures? So Haemophilus influenza, B. pertussis, Pasteurella, Brucella, etc. In case of bacilli, that is rod, gram negative rod. So very simple examples of gram negative rod are Shigella, Pseudomonas, E. coli, Klebsiella, etc., etc. Now even these are the most widely used cultures for your practicals in your college levels, so those are only listed here. Okay, but there are different genus or genera present on the group of gram negative rod shaped aerobic bacteria. So what about those? Okay, so you don't know what is growing on your plate, so you have to consider all and you have to follow Bergey's manual for biochemicals. Okay, so that's how you have to proceed. Now let's take one example here. So suppose we have a culture which is gram negative rods, okay, and it is aerobic because I have just incubated it in a incubator and in aerobic conditions, so it is growing well, nice, flourishing. So I have that culture and now I have to identify it. So how to proceed? So I have isolated, I have done my colony characteristics, I have done gram staining, it is, the result is gram negative. I have done the motility and it is motile. Okay, suppose it is motile and it is rod shape. So this is my first report of colony characteristics which is ready. Now I will open Bergey's manual. I will go to the groups and I will see what groups are there which matches to my report. Okay, so fourth group is what I could match my results with. So I will open this fourth group and I will see what all genera or what all genuses they are described here. Okay, and I will move further for biochemicals. Now you can see a whole table is given here with different biochemical tests. Okay, not just biochemical test, you can see starting from the cell shape, okay, gram staining, to cell shape, to the cell diameter, everything is mentioned in this one particular table. Okay, so this table is titled as general characteristics of genera of gram negative aerobic eubacteria rods and cocci. So you can see this is just one table which has around 7 genus. Okay, for comparison, there are further 6 to 7 pages which has all the uh, genus mentioned for the comparison, but I cannot cover everything in just one video, so I am taking these as example. So let's assume that my culture is acetobacter. Okay, so what I will do, I will start performing the biochemicals and then I will compare my results. Okay, so you can see here the given genus is, first is acetobacter, then acidiphyllum, then acidomonas, then acidothermus, then acidoverax, and further on acinetobacter, etc. So you have to perform these biochemicals and then you need to report your results and open again your Bergey's manual and compare your results. Okay, which biochemical test gave you positive results, which biochemical test gave you negative results, and you need to compare and you need to identify that yes, my results matches to say this particular genus, that is Acinetobacter. So my culture belongs to genus Acetobacter, not Acinetobacter. So my culture belongs to genus Acetobacter. Okay, so you can read this description to confirm your results. And further, if you want to identify the species of genus Acetobacter, then you can see here tables like this are given in Bergey's Manual of Determinative Bacteriology for further identification of these species. Okay, so again the list of biochemicals is given which helps you to identify between different species of a particular genus. Okay, so you can see here Azotobacter, for Azomonas. There are different tables which are given which helps you differentiate between the species of a particular genus. Okay, so that's how we use Bergey's manual, okay, to identify our culture. Now you have identified your culture. Now the last thing you have to do is write a report. Okay, so many institutes or many colleges, they have a format of or a sheet where a particular format is given and that is followed. So this can be, say for example, so this is just for reference. Okay, a report sheet format can vary. Okay, so you can see here gram reaction, that is gram strain result you have to write, then microscopic morphology and arrangement, that means what you have observed under your microscope. Okay, then description of colony, color, then you can see other colony characteristics like colony shape, type of margin, elevation, and others. You can mention what you have observed. Then growth characteristic. Okay, so to what temperature and for what time you have incubated your culture, it requires oxygen or not, that means it is aerobic, facultative, or anaerobic. Then oxidation fermentation test. Okay, you have to mark that. Then biochemical test, now this may vary depending on the group you are referring to, but there are around 10 to 15 biochemical tests that you have to perform at least to identify your culture. You cannot just perform 2 to 3 biochemical tests and say that yes, my culture belongs to this particular genus. Okay, that's actually a wrong method. You have to go through the list and try to perform at least 10 to 15, okay, even 20 biochemical tests. Okay, it may happen that some of the reagents or some of the material which is required for a particular biochemical test is not available in your lab. Okay, so you can omit that, but it doesn't mean that you have to omit all, okay, and do the easy biochemical test. That's the very wrong approach. You have to minimum try to perform 15 biochemical tests, okay, and other sugars for fermentation, oxidation test, etc., etc., that you have to report. And other than biochemical test, do you find any other test that you think is important for this particular culture and you have performed that, then you have to write about that test here. And then you can identify, you can write that my, according to Bergey's manual of determinative bacteriology, my culture may belong to genus this and species this. Okay, so we had our teachers made us write the word may belong to, okay, because we are working on a college level lab, so it may happen that human error may cause some test to come positive and you may slightly get results here and there and you identify a wrong genus, then it may cause a problem. So our teachers made us write that may my culture may belong to genus. Okay, never directly write that my culture belongs to this particular genus. Okay, you have to do some uh, sequencing also. If you are doing research work, then you have to be double check, you have to cross check your results by doing molecular study. Okay, so that's the other another way of identifying your culture. And yes, so this was all about identification of unknown bacterial culture, and I hope you have now a clear approach like how to proceed and what to do. So I hope you like this video. Do share these videos with your friends and do subscribe to my channel and keep supporting. Thank you.